Hello everyone, we are going to describe about the transduction phenomenon and how the generalized transducing particle is produced and the experimental approaches regarding to generalized transducing event. So what is transduction? Apart from bacterial conjugation and transformation, a bacterial DNA can be transferred from one bacterial cell to another cell by a phage particle containing bacterial DNA, and such a particle is known as transducing particle. So phage is a vector which can carry a bacterial DNA from one bacterial cell to another. Two types of transduction or transducing particles are actually present. One is called generalized transducing particle and another is known as specialized transducing particles. Generalized transducing particles contains only bacterial DNA. However, the specialized transducing particle contains both bacterial DNA and phage DNA linked within a single DNA molecule present within the phage head. So in this video we are going to describe about only the generalized transducing particle, how the generalized transducing particle is produced and how recombination will occur. So this is the normal process of how the phage infects a bacteria. So say this is the phage, any kind of phage, say phage P1 or phage P22, which upon infection a bacteria, upon infecting a bacteria, what will happen? This is the phage DNA, say red colored, this is the phage DNA. The initial event after infection of phage to the bacteria is the degradation of the bacterial whole genome. So this is the degradation of the bacterial DNA. So phage eventually produces a nuclease which causes the destruction of the entire bacterial DNA, so that phage can use the DNA as a source of nucleotide to make their own DNA. So this is the phage DNA. So this is the one copy of the DNA which was initially injected by the initial phage, and they produces many copies of the phage DNA now using the bacterial DNA, and after that they need to package to produce the infective virion, and the virion will ultimately lyse. So upon infection, the initial event is the degradation of the bacterial DNA. We have to remember this thing, that is the after infection bacterial DNA will be degraded by the phage nuclease, and the degraded DNA will be used as a source of nucleotide to make their own progeny phages, so that the progeny phages need to be packaged. And now how the generalized transducing particle is produced. Imagine the phage is P1 and this is the E. coli bacteria having the leucine gene, leucine plus, that is leu plus gene in its chromosome. So when the phage DNA gets injected or incorporated into the host cell cytoplasm, the initial event is the degradation of the bacterial DNA. So the bacterial DNA will be degraded by many segments actually, and the P1 phage will produce many particles by using the DNA as a source of nucleotide. But during packaging, if it makes a mistake, that is, during packaging, if one of the segment of bacterial DNA remains in the cell cytoplasm is comparable to size of the P1 phage, then mistakenly a P1 phage can carry the bacterial DNA instead of the phage DNA its own, right? So phage can make a mistake. When it makes a mistake, if the size of bacterial DNA is comparable in size to P1 DNA, in that case it may be packaged into the phage particle instead of the P1 DNA. The positions of the nucleus cut in the host chromosomes are fairly random, so a generalized transducing particle may contain a fragment derived from any region of the host DNA, and a sufficiently large population of P1 phage will contain, this is a sufficiently large population of P1 phage which contains particles, at least one of which carries host genes. About 1 particle per 10 to the power 3 progeny is a transducing particle. So this is the transducing particle, and these are normal viable phages. So normal viable phage normally packages its own DNA, but a transducing particle mistakenly takes up the bacterial DNA if the size of the bacterial DNA fragment is comparable to size of the P1 DNA, and phage can mistakenly takes up the P1 DNA, bacterial DNA. In that case, a phage will be produced that will be known as generalized transducing particle. Why generalized? Why not specialized? Because the segment of DNA can come from any region of the bacterial chromosome. Not essential the leucine gene will come. Any other genes present in any other part can be taken by the phage particle. So consider a situation that in this case the donor bacteria was leu plus, and somehow the transducing particle is produced that carries the leu plus gene. So when this particle next infects another bacteria, the first event that the bacterial cell will not lyse. First event that the bacterial cell will not lyse, because this is the transducing particle entirely contains the bacterial DNA. So the bacterial DNA if the bacterial DNA contains the leucine gene, in that case it will inject the leu plus. This one is leu plus basically, and this is the leu minus DNA. So if if the transducing particle infects a Lu- recipient bacterium and incorporates its Lu plus gene, then a double recombination event will successfully causes the generation of a Lu plus recombinant or Lu plus transductants. So Lu- fragments will be degraded, will be excised out and it will be degraded. So this is the basic way by which a generalized transducing particle ultimately forms a recombinant. About one particle per 10 to the power 3 progeny will be transducing particle, and the idea is about 1 out of 10 to the power 6 progeny bacteria will be a Lu plus transductants. The next is the experimental approaches to generalized transduction. Transduction was originally discovered by using FASP22, and the bacteria for infection was used was Salmonella typhimurium. And this experiment used the DNA density leveling technique. Host bacterium, here the host bacterium, that is say Salmonella typhimurium, is labeled with N15 by growing for many generations in a medium containing N15. So the bacterial DNA, the host bacterial DNA, Salmonella typhimurium DNA, is labeled with N15, and it is infected with a N14 P22 phages. So it is infected with N14 labeled P22 phages, and then the phage bacterium complex is grown in the same N14 medium. So the phage is labeled with N14, and the medium where the phage bacterium complex is placed, it is also N14, but the bacterial DNA, initial bacterial DNA, is labeled with N15, right? So because the phage bacterium complex is growing in N15, so all the viable phages, all the progeny viable phages will be labeled with N14, because the media is labeled, media contains N14, so all the progeny phages will be N14. But if a phage, if a P22 phage mistakenly takes up the bacterial DNA which is labeled with N15, then a transducing particle will be labeled with N15. So these are the transducing particle which may occurs once in 10 to the power 3 phages. The transducing particle will be generated here, and the transducing particle will be labeled with N15. So if a phage mistakenly takes up the N15 labeled DNA, then the phage is a transducing particle. So the phage's transducing particle must be labeled with N15 label, while the viable phage will be labeled with N14, because initial phage was labeled with N14 and they were growing in a medium that is also N14. So all the progeny viable phages will be labeled with N14, only the transducing particle will be labeled with N15. So what we can do, we can separate these viable phage and transducing particle by cesium chloride density gradient ultracentrifugation, which separates the particle by density. The centrifuge tube, which contains both of these viable phage and transducing particle, can be punctured by tube bottom, at the tube bottom and collecting successive drops, and each fraction was tested for the presence of both viable phage and transducing particle. And the viable phage should be labeled with N14. This one, the orange colored, actually orange labeled, orange color, which denotes the N14 labeled viable phages, and N15 labeled transducing particle will get, we can get a peak for the N15 labeled transducing particle. So the viable phages had the density of particle containing N14 level DNA, and the transducing particle will have a particle density of N15. So if we generate this kind of peak for N15, then we can surely say this peak is actually due to the presence of transducing particle. Now there is a question that why this peak, why the viable phage peak is not at just N15, but just before of that of the N14, but just before of that of the N14. This peak should be drawn by that point, this peak, but this is came before that point, that is it should contain some N15 level DNA or nucleotide. Why? Because there is a small shift to the higher density resulting from incorporation of some N14 level nucleotide obtained from the degraded bacterial DNA. So we know that the phages are growing in N14 medium, but they can use the bacterial DNA as a source of nucleotide. So if some of the bacterial DNA gets incorporated, some of the bacterial nucleotide gets incorporated to make the progeny phages, in that case the progeny phage will not get totally N14, but at least 2% or 1% N15 also. So that is why the peak appears here, right? So this is the mechanism, and now we are going for the co-transduction phenomena. Now the idea is that consider a population of P1 prepared from Leu, Gal and Bio plus bacterium. So we are taking Leu plus, Gal plus and Bio plus bacterium. The sample will contain particles able to transfer any of these genes to another bacterium. But the idea that if say this is the recipient cell contains Leu minus Gal minus gene, and another recipient cell which contains Gal minus Bio minus gene. So we can see there the Leu and Gal genes are separated far apart, while the Gal and Bio genes are very closely present. So the idea of co-transduction is that two genes can be simultaneously gets incorporated within the same cell if they are present very closely. That means you cannot generate a Leu plus Gal plus particle simultaneously. You can generate Leu plus transducing particle, you can generate Gal plus transducing particle, but you cannot generate Leu plus Gal plus transducing particle simultaneously, or if it is produced, the recombination frequency will be very, very low because the size of the Leu plus Gal plus transducing particle is very large compared to that of the head of the P1 phage, and that is why it will not package rapidly. Okay, so that is why you can generate either Leu plus transducing particle or maybe the Gal plus transducing particle. If you generate Leu plus transducing particle and infect a bacteria that is Leu minus Gal minus, in that case a double recombination event will generate a Leu plus Gal minus bacteria only. Simultaneously, if you generate a say Gal plus, if you generate a say Gal plus DNA, this is the situation, that is if it generates a Gal plus DNA transducing DNA containing galactose gene only. If it is galactose gene, then a recipient bacteria which is Gal minus and Bio minus, a double recombination event will ultimately generate Gal plus Bio minus. So either Leu plus Gal minus will be generated or Gal plus Bio minus will be generated, but you cannot generate Leu plus Gal plus simultaneously. This is the scenario because leucine and galactose genes are very far present. However, the Gal and Bio genes are very closely present and they are separated by about 2.3 in 10 to the power 4 base pair, and the P1 phage can contain a segment of DNA that is about 8.5 in 10 to the power 4 base pair. So we can compare these two values. The P1 phage contains 8.5 in 10 to the power 4 base pair in its head. However, the Gal Bio genes are separated by only 2.10 to the power 4 base pair. So there is every possibility that a P1 phage can carry simultaneously Gal and Bio genes. This is the case. Definitely it can carry only galactose gene, or it can carry only biotin genes. If it carries only galactose gene, then it will generate Gal plus Bio minus. If it carries only biotin genes, then it will generate Gal minus Bio plus, because nucleus can sometimes be cut mistakenly between the Gal and Bio genes. But if it produces a fragment that contains both Gal and Bio genes, then ultimately you can generate a Gal plus Bio plus particle. So co-transduction will be formed. It is a minor species, but these are the major species because these are the single recombinant, and the double recombination frequency will be always lower than the single recombination frequency. So the idea is that co-transduction, in order to get co-transduction, what you will require? You require two genes which are very closely present. Say Gal Bio can be co-transduced, but Leu Gal cannot be co-transduced because leucine and galactose genes are very far away. So it will not be carried within the same P1 phage. But if the Gal and Bio genes are present, they can be carried within the same phase. So we can generate Gal plus Bio plus transductance, but we cannot generate Leu plus Gal plus transductance. Either Leu plus Gal minus, or it may be Leu minus Gal plus will be generated. That is not described here, but there will be. You can surely understand the fact that you can generate the Leu minus Gal plus bacteria also, right? Hope you understand.